thank you very much for this invitation here. This is my first AbSciCon, and I'm enjoying it very much. Before I'm going to tell you about the research we have been doing in the lab, I first want to mention two thoughts on the Rna World hypothesis, because over the last days, a number of people have expressed disappointment or negative feelings against the Rna World hypothesis. And I want to clarify two things. No laughing. I want to clarify two things. So first of all, we have pretty good evidence from ribosome, tRNA, RNAsP, and nucleotide cofactors that something like an RNA world existed as an earlier stage in evolution. It probably was not the first stage of life. And probably, or most people, I think, agree with me that there probably was not a pure RNA world, but something like an RNA world existed as an earlier stage in life. And the other point I want to make is the worth of a hypothesis is not only how well it describes reality, how accurate it is, it also is to what kind of experiments and tests does it give rise to forward our understanding. And I think in this respect, the Rna World hypothesis has been exceptionally fruitful. Now, we can discuss this. I'm happy to discuss this. I want to also mention that this is one example of a conceptual question that is very important to address, and that we are addressing in these research coordination networks. I'm part of this PC3, Prebiotic Chemistry and Early Earth Environments. And Lauren Williams sitting here, he's one of the head of that group. And I encourage you to get in contact with that group because NASA is stimulating that network and we may have a lot of fruitful outcomes from that. So enough for that. I want to now go to the research that I have been doing, which is focused on the Rna world. When I think about the Rna world, I'm thinking about this overall picture here, where by definition we must have used prebiotically available compounds to generate nucleosides. How they were generated, I don't know, and there's a number of good organic chemists that are addressing this question. What my group has been working on mostly is the activation or the generating of activated nucleotides from these nucleosides. And after you have chemically activated nucleotides, you can form by RNA polymerization RNA polymers. And I want to emphasize this addresses a mature RNA world. This might not have been the case in an early RNA world. Please do not take pictures. Almost everything I'm showing you is unpublished. So I'm not too concerned about this picture, but about the other slides. We are focused on this reaction here, and Leslie Orgel showed in 1978 that if you incubate adenosine with trimetaphosphate at a pH of 13, then this will give rise to ATP, to a 5' triphosphate. Within a few hours, about 5% are converted to ATP. And what this shows that in general this reaction is possible. However, a pH of 13 is incompatible with an RNA world because everything degrades within minutes or even seconds. What we did in my lab in previous years is we have generated catalytic RNAs that react trimetaphosphate with their own 5' hydroxyl group. So you're making RNA 5' triphosphates, which shows that in general the chemistry works and that this reaction can be catalyzed by RNAs at neutral pH. However, what you eventually want to have is nucleoside 5' triphosphates, because in every single living organism we know these are the central metabolic molecules, and especially in an RNA world. So what is the problem? Why haven't we developed a ribozyme that makes free nucleoside triphosphates? There's two general difficulties. One of them is from the ribozyme side. This ribozyme needs to bind two small molecules and catalyze the reaction. And that is a tall order to ask from an RNA. And the second one is For in vitro selections, you always need to tag your pool molecule, your successful pool molecule, with some handle so that you can later pull on that handle and pull that molecule out of your population. If your successful product is a freely diffusing small molecule, then this is hard to do. And in the next slides, I'm going to show you how we were addressing these challenges. First of all, this here is a pool molecule with 150 random nucleotides. That's the pool length we used. And the reaction we want to catalyze is here 6-thioguanosine, a nucleoside that reacts with trimetaphosphate to form 6-thioguanosine 5' triphosphate. And I will tell you in a minute why we have these thio modifications here. So this problem how to tag pool molecules that successfully carried out a reaction, we addressed by attaching a polymerase ribozyme, or by base pairing a polymerase ribozyme to the 3' end of the pool, so that polymerase ribozymes could take 6-thio-GTP that was made by these ribozymes and tag it to their own 3' end. And because the wild-type polymerase ribozyme is not very good in using 6-thio-GTP, we performed an in vitro evolution experiment that optimized this polymerase ribozyme for the attachment of 6-thio-GTP, and we found 4 mutations. 2 of them have a 2-fold effect, 1 of them has a 50-fold effect. So this ribozyme is 200-fold better in attaching the 6-thio-GTP to the 3' end of the pool than the original wild-type ribozyme, and that proved to be good enough for us. Then we have these pool molecules with these 3' tags of 6-thio-GTP or 6-thioguanosine, and to separate the successfully tagged pool molecules from those that were not tagged, we used aminophenol mercury polyacrylamide gel electrophoresis, which was first used by Peter Unrau in an in vitro selection as the selective step. So once you have these tagged pool molecules, you run them on these polyacrylamide gels, only those that are tagged are stuck on this APM interface, and then you can cut out these gel pieces and use them for your next step. So this is the first challenge that we overcame, how to tag these successful pool molecules. The second challenge is one next to tag inactive pool molecules, how do you avoid tagging inactive pool molecules? Because you have to keep in mind, if this here is an active pool molecule that generates 6-thio-GTP and that hopefully leads to the tagging of its own 3' end, you can also have the 6-thio-GTP now diffusing around to a neighboring pool molecule and lead to the 3' tagging of that pool molecule. And to prevent that from happening, you need to compartmentalize the system. And we did this in the form of an emulsion where each pool molecule was within its own compartment. And I believe that's the first selection that does that from completely random sequence, that we compartmentalize each pool molecule. So the system we used is an emulsion formulation that was shown by Phil Holliger's lab in 2004. We used the emulsifier Able EM90 in heavy mineral oil. And then we used a machine called the microfluidizer from Microfluidics. I have no association with the company. And it basically presses the raw emulsions through a cell with a defined shearing force so that you end up with a very homogeneous size distribution of your droplets. And in collaboration with Doug Magdy at UCSD, we measured the diameter of these droplets by dynamic light scattering. And you see from the nice single exponential curves that we got a pretty homogeneous size distribution with a diameter of 150 nanometers. And this diameter is significant because if you imagine an aqueous droplet with a diameter of 150 nanometer, then if you take a solution of molecules with a 1 micromolar concentration, then on average each droplet has 1 molecule per droplet. So we are going to the molecular scale in terms of separating out our pool molecules, and that's important, that's essential for the selection. So after My graduate student, Arvin Akupi, spent 3.5 years of hard work on establishing this selection system. He was ready to do the selection, and the selection was done in about half a year, and the analysis in another few months. So first, I want to quickly show you the selection system together. First, we start with these DNA pool molecules that are then transcribed in RNA pool molecules, and Arvin had an effective complexity of 1.6 times 10 to the 14 molecules. Then they get compartmentalized in these emulsion droplets together with the polymerase ribozymes with 6-thioguanosine and trimetaphosphate, in the hope that some of them generate 6-thio-GTP and are then tagged at their 3' end. Then the emulsion is broken open, all the aqueous content is released and thrown on aminophenol mercury page. We process these molecules by reverse transcription, PCR, and then complete one cycle. Arvin went through 18 cycles, and you see that after 10-12 cycles, he got an exponential enrichment. He increased the stringency of the selection to select for the most efficient ribozymes. And after this was done, he did a high-throughput sequencing analysis, where in the end, the population was dominated by 5 clusters. And then Arvin had to quickly finish his PhD and go into biotech industry. Josh Areola, my new grad student, he took over for the biochemical characterization. And this is how we did the biochemical characterization. So the randomized portion of these pool molecules is incubated with 6-thioguanosine trimetaphosphate to generate 6-thio-GTP. And then a short 10 nucleotide long radio-labeled RNA is attached to the polymerase ribozyme to serve as the reporter whether 6-thio-GTP was generated. We screened clones, between 2 and 5 clones per cluster, and you see that cluster 5 gave the strongest signal. And here you see a kinetic analysis of this cluster, where this is the negative control, no ribozyme present. This is the positive control with only 6-thio-GTP. And here you see the reaction catalyzed by this ribozyme. And with this, I just want to acknowledge Arvin Akupi, who did establish the selection and did most of the work, Josh Areola, who is currently doing the biochemical analysis, Doug Magdy, who did the dynamic light scattering analysis, Brian Pagel for helpful discussions on the emulsions, and NASA for funding. Thank you for your attention.
Thanks very much, Julie. I can really appreciate having one student work really hard to generate the system and someone else having to come in and then follow it up to do the biochemical analysis. Yes, Peter.
Yeah, so really exciting work. Do you think that these ribozymes that you selected, they presumably, they make 6-thio-GTP, but they also might have adapted themselves to the polymerase. Do you have any evidence that either is true or not true, that if you just feed 6-thio-GTP into the system, you have different abilities with polymerase to actually add the 6-thio-GTP to the substrate? And have the ribozymes that you've evolved actually evolved some sort of affinity for the polymerase itself?
Yes. So of those clones I showed you, you notice that the most enriched cluster 1 and 2, they are not as active in this specific assay. And in this specific assay, we were detaching this randomized region of the pool from the annealing region to the polymerase ribozyme. So this cluster 5 seems to do well. The other clusters are more active in a different assay. So the answer to your question is yes. Many of these pool molecules seem to have developed a dependence. We will probably go with this ribozyme because optimizing and cutting down to the catalytic core of a ribozyme that is dependent on another 200 nucleotide ribozyme is a pain. And we would like to have a small ribozyme that hopefully can be optimized to be highly active. And so your follow-up question was with canonical nucleoside triphosphates, is that correct?
No, I think you answered everything. Just what kind of turnover are you actually seeing?
What kind of?
Turnover are you actually seeing?
The turnover is currently quite low. We haven't really characterized that yet. It's, I think, less than 10 at this point.
to stay on time, why don't we move on? Thank you very much, Julie.